Hello children, today we are going to see about the book back questions and some inside questions of unit 2 lesson 4 principles of inheritance and variation. Now the first 2 mark questions, some are in the textbook itself, we are covering those things and also the internal questions also. So first question is, what are Hollandric genes? So Y-linked are called as Hollandric genes. The genes present have the differential region of Y chromosome, and the Y-linked genes inherit along with the Y chromosome and phenotypically express only in the male. So if you write these 2 points, you will get full marks. The second one is, what is haplodiploidy? So it is a sex determination mechanism which is commonly seen in hymenopteran insects. So in this sex of offsprings is determined by the number of chromosome sets it receives. So for example, in honeybees, the fertilized eggs are called as diploid eggs and they become female, and the unfertilized eggs are called as haploid and they become the male bees. Mention the symptoms of Down syndrome. So Down syndrome is also called as Mongolian syndrome. So the symptoms of Down syndrome is called as, it has severe mental retardation, defective development of central nervous system, flattened nose, ears are malformed, mouth constantly open and the tongues are Now what is karyotyping, the fourth question? So it is a technique through which a complete set of chromosomes is separated from a cell and the chromosomes are arranged in pairs. So this is called as karyotyping. So this is a type of inheritance in which the recessive linked sex linked character, that is X of father, is given to daughter and then it is passed through from daughter to her sons. So this is known as crisscross or sex linked or X linked inheritance. So if they ask any of these things also, you have to write this inheritance. So you have a flow chart of a diagram here given, so that you can remember very easily. So it is X chromosome of father which is transferred to the daughter, and then the daughter has 2 X chromosomes, out of which the father's X chromosome will be going to the son. So that is his grandfather. From the grandfather, the grandchild will be attaining the character. So this is called as the crisscross inheritance. The sixth one is, why are sex linked inheritance characters are more common in male human beings than females? So the sex linked recessive characters are more common in males than in females because males are hemizygous. This word you have to mention, otherwise you will not get marks. So males are hemizygous and therefore express the trait when they inherit one mutant allele. So these are the highlighted colored ones or important words which we expect. Draw a schematic diagram to show the marriage between a colorblind male and a normal female which result in a normal individuals in the F1 generation. So if you see the flow chart like, so the normal female will be having the XX chromosome and the colorblind male will be having XC and Y. So it's a XC is nothing but the X chromosome is showing the gene colorblindness gene. So when we take the gametes of the parents, you will get X plus, that is the common female is represented as X positive, that is no infection or any disorder in them, and XC is the colorblind gene and Y for the male inheritance. So if you take the cross of between the normal female and the colorblind male, you will be getting a daughter with normal but she is a carrier, and a male with normal chromosome if he gets the X chromosome from the mother. What do you mean by pedigree analysis, and represent it in a diagrammatic form? So it is a study of traits as they have appeared in a given family line for several past generations. So that is called as the pedigree analysis. So if any disease is running in the family, it will be shown for several generations as it is transferred to one parent to the other children in the next generation. So, you can represent the diagram like this. The square represents the male here and the circle represents the female here. When they get married or mating happens, you will be getting one daughter and one son. So, that is how you represent a pedigree analysis. So, if you define pedigree, so pedigree is a family tree drawn with standard genetic symbols showing the inheritance pathway for specific phenotypic characters. So, that is pedigree analysis. So, the word you have to mention here, it is the family tree and it is a standard genetic symbol. Using the standard genetic symbols, we are drawing the pedigree. One, what are the symptoms of phenylketonuria? So, it has severe mental retardation and light pigmentation of skin and hair, that is almost they will be fair in color or white in color, and phenylpyruvic acid is excreted in the urine. So, these are the symptoms of phenylketonuria. Define multiple allelism. So, when there are 3 or more alleles of a gene control for a particular trait, it occupies the same locus on the homologous chromosome of an organism. So, they are called as multiple alleles and their inheritance is called as multiple allelism. So, example for this is in our textbook, it is ABO blood group. Define genetics. So, genetics is a branch of biology that deals with the study of heredity and variations. So, it describes how characteristics and features pass from one generation to another generation. So, that is called as genetics. So, if a person is having a group A blood, then write the possible genotypes and antibodies in his blood. So, group A, if you are having a blood group A, then the genotypes will be of 2 types, that is IAA or IAIO. So, antibodies of this they will be having B type antibodies, the opposite one will be the antibody. So, if you are having B group, then you will be similarly it will be IBB or IBO, and he will be having the antigens A, antibodies A. Pars. So, pars is pseudo autosomal regions of Y chromosome and they are present on both ends of the Y chromosome. So, the synapses and recombination takes place during meiosis with the X chromosome in these places only. So, this is called as pseudo autosomal regions. What is euchromatic gene? So, it is a functional genes present in the non-combining region of Y chromosome and it is also called as NRY, and it has a gene of its region and it is called as sex determining region called SRY, and which produces the testis determining factor, the TDF, present in the male testis. So, this is the euchromatic. What is barred body? So, the condensed part of the sex chromatin first found in the nerve cell of a female cat and then it is absent in the male cat of the nerve cell, and this is called as barred body by Barr and Bertram in 1949. What is ideogram? So, ideogram is a diagrammatic representation of karyotype showing all morphological features of the chromosome grouped on the basis of their position of centromere. So, that is very important. It is based on the position of centromere and the order of a series of decreasing size. Chromosome, it helps in chromosome banding and painting technique and it is used to identify each chromosome. Genetic disorder and mention its type. So, a genetic disorder is a disease or a syndrome that is caused by the abnormality in the individual DNA. So, in the DNA level, if there is any disorder, then we call it as a genetic disorder. So, genetic disorders are of 2 types, that is Mendelian disorders and chromosomal disorders. The missing boxes. So, you have the reactions in your textbook. So, they will be giving some blank like this and they will be giving one word. So, you have to fill those things. So, here the question is tyrosine is given, other 2 are not given. So, the answer for this is phenylalanine in presence of phenylalanine hydroxylase gives tyrosine. So, this is the reaction. You have to learn only 2 reactions you have in your textbook. So, it is very easy to remember. 3,4-dihydroxyphenylalanine, that is DOPA, gives what in presence of what enzyme? So, it is 3,4-dihydroxyphenylalanine DOPA in presence of tyrosinase gives melanine. So, this is the answer. You have it in your textbook. So, you have to read the textbook very carefully. What is Cooley's anemia? So, the beta type thalassemia is also called as Cooley's anemia, and there is increased production of alpha chain and damages the membranes of RBC. Now, 21st is what is heterogametic sex determination. So, when one of the sexes produce similar gametes and others produce dissimilar gametes, that is called as sex determination. The sex of the offspring is determined at the time of fertilization. So, there are 2 types, that is heterogametic males which has XXXY or XXXO, and heterogametic females which are represented as ZOZZ and ZWZZ. So, here the first term indicates the females, that is the heterogametic males are XY and XO, and heterogametic females are ZO and ZW. What is Mongolism? So, the Down syndrome is formally called as Mongolism. It is not given in the textbook, but it is commonly known as Mongolism. So, it is a congenital disease caused due to the trisomy condition in chromosome number 21. A person is diagnosed with gynecomastia. So, write the conditions why it occurs and what is it called as. So, gynecomastia is a genetic disease in which male breasts are enlarged feebly, and it is also called as Klinefelter syndrome or XXY males, in which they have 47 chromosomes instead of 46. So, you can split that one as 44 autosomes plus XXY. So, you will be having an additional X in the male. So, that is called as Klinefelter syndrome, or this causes gynecomastia in return. So, that is the difference in the chromosome number, which is find CDE nomenclature. So, you can write this same question, same points even for 2 marks, 3 marks or 5 marks also. So, CDE nomenclature is nothing but it is first given by whom we have to write, that is Fisher and Race, and it is used to determine the RH factor, whether the person is positive, RH positive or RH negative. So, it involves 3 pairs of RH alleles, that is capital C, capital D, small d, and capital E and small e. So, at different loci on a homologous chromosome pair, and these 3 genes are linked. So, that is main important point you have to write. Now, in these genes, the C and E genes are codominant, that means they equally express when during their phenotypic character. So, in the genotype carrying capital D, then it is called as a dominant gene, and the person who gets the D is called as RH positive, and the person who receives small d, that is the recessive gene, then he is called as RH negative. So, for example, you can write like CDE or all capital letters, or one small c and one capital D and one small e. So, that these 2 here, since the D is dominant here, the person who has D in his blood group genotype, he will be getting RH positive, and the person who gets a recessive D type, even though there is capital C and capital E or recessive C or recessive E, the person is declared as RH negative because of the gene D. So, this is the diagrammatic representation of the CDE nomenclature or Fisher hypothesis. Everything is the same question. So, do not confuse. So, if you read carefully, it is there in their textbook. So, what is CDE nomenclature means? Fisher and Race hypothesis is also called as the CDE nomenclature. How did Wiener explain the RH factor or explain the Wiener hypothesis? So, anything they can give the question. So, first we have to write the scientist's name, or he is Dr. Alexander Wiener, and it is why it is used. It is usually useful in the describing the phenotype of the blood group, and the RH gene occurs at one RH locus and it has 8 multiple alleles. So, there are 8 major alleles called as R0, R1, R2, RZ, R, R dash and R double dash and RY. So, all genotypes carrying the dominant R allele are called as RH phenotype, and the one double recessive genotype, that is small r, they have RH negative phenotype. So, these same points also you can write for even 2 to 3 marks also. So, this is a simple diagrammatic representation of the Wiener hypothesis, in which this green one is the chromosome and the yellow point, this is called as the location of the gene or the loci where the RH gene is present. So, in this region you will be getting either one of the allele on this spot. So, that can be either dominant R or the small r. So, what is trisomy condition? Mention any 2 diseases associated with it. So, the presence of 3 copies of a chromosome rather than 2 normal is called as trisomy conditions. So, what instead of 2 normal chromosome, it is having 3 copies. So, that is called as the trisomy condition. So, here we have 2 classical examples for trisomy, that is the Mongolian Down syndrome. It is affecting the chromosome number 21, which has 3 pairs of chromosome 21, and the second one is Patau syndrome and the trisomy 13. Define sex-linked inheritance with some examples. So, the inheritance of a trait that is determined by a gene located on one of the sex chromosome is called as sex-linked inheritance. So, examples of sex-linked inheritance is hemophilia, color blindness and muscular dystrophy. The 28th question, what are the causes of chromosomal abnormalities? Mention a few examples. So, chromosomal abnormalities arise due to chromosomal non-disjunction location, deletion, duplication and inversion. So, these are the abnormalities, that is non-disjunction means the chromosome will not move during the after metaphase stage to the poles of in the anaphase stage. So, that is called as the non-disjunction. Translocation, you know that the genes will be moving from one place to another place, that is called as translocation, and deletion means either of the gene will be deleted, then that is called as the deletion, or duplication means the same gene will be copied again, then it is called as duplication, or inversion means the series will be changed, that is if it is ABC, the inverse will be C, B and A. So, that is called as the inversion. So, these are the some of the examples for this chromosomal abnormalities are Klinefelter syndrome, Turner syndrome, Down syndrome and Patau syndrome. So, these are the 4 examples you have in your textbook. So, if you have to know all the characters of these diseases and what is the chromosome affecting this, what are the causes of Mendelian disorders. So, mention few examples. So, alterations or mutation in a single gene causes Mendelian disorders. So, Mendelian disorders can be also caused if it is change in single gene, then we call it as a point mutation also. So, point mutation or a single gene alteration causes Mendelian disorders. So, some of the examples are thalassemia, albinism, phenylketonuria and Huntington's chorea. The 30th question, write the genotypes of the following organisms. So, you should know the genotypes of the following organisms. So, cockroaches, Drosophila, domestic chickens, fishes are given here. So, you have many more examples also, you should remember those things. So, in this they have asked cockroaches. So, if you know, the cockroaches and Drosophila are the heterogametic males, and cockroaches, that is XXXO, they have the genotype of XX and XO, and Drosophila you have XX and XY. So, these are the heterogametic males. So, the XO and XY are the male gametes here, and heterogametic females. So, domestic chickens have ZO and ZZ type, and fishes they have ZW and ZZ. So, these are the genotypes of the following organisms. Describe the Y chromosome they have asked. So, you have to write who has discovered this Y chromosome. It was Stevens in 1902, and you have to write the shape of the chromosome. That is, it is acrocentric chromosome, and this is in the do you know box in your textbook. So, it is of 60 MB in size and with 60 functional genes, and Y chromosome is largely heterochromatic, and heterochromatin is a condensed and it is inactive chromatin. Describe the X chromosome. So, X chromosome is discovered by Henking in 1891, and it is a metacentric chromosome, and it is 165 MB in size and with 1000 genes. And since there are 2 chromosomes in females and 1 in males, X in females is inactivated and it is called as bar body. So, we have in females we have 2 X chromosomes in females and 1 in males. So, the X which is in double, this is also called as dosage compensation. So, where the female is inactivated and it is called the bar body. What is N-1 rule? So, it is a formula to know the number of bar bodies present in a cell. So, where N is the total number of chromosomes present. So, the number of bar bodies is 1 less than the number of X chromosome. That is, you have, if your males are there, you have XY. So, here the X is only 1. So, this X is the, if you take the formula N-1, so where you will have the number of X chromosomes. So, here the X chromosome is only 1, that is active X is only 1. So, there is no bar bodies, and females you have bar bodies, 2 X are there. So, you will be having 1 bar body and 1 X active X. So, that is the N-1 rule. Now, let us go to the 3 marks questions now. Write the applications of karyotyping. So, you have 4 to 5 questions in them. The first one is, it helps in gender identification, and it helps to identify the abnormalities and aberrations of chromosomes like aneuploidy, non-disjunction and duplication, etc. Here I have combined the 2 marks and you can write it, or you can elaborate that one into 2 points also as it is in your textbook. So, it is also used in predicting the evolutionary relationship between the species, and the genetic diseases in the human beings can be detected by this technique. So, these are the 4 points which are there for the applications of karyotyping. So, it helps in gender identification, and it helps to identify the abnormalities and aberrations of chromosomes like aneuploidy, non-disjunction, duplication, etc. And it is also used in predicting the evolutionary relationship between the species, and genetic diseases in human beings can be detected using this technique. This is what is lionization or lion hypothesis, both are same. So, it is a condition where 1 copy of gene of chromosome is inactivated in some female mammals. So, what is lionization? It is a condition where the 1 of the copy of the chromosome, that is usually the female X chromosome, is inactivated in some female mammals. So, the inactive chromosome becomes highly coiled and it is called as the heterochromatin. So, the heterochromatin is also called as the bar bodies. Distinguish between heterogametic and homogametic sex determination. So, heterochromatic sex determination. So, it is a two type of gametes are produced here, and if you see in the homogametic sex determination, there is only one type of gamete produced there. So, homo means similar or single type, hetero means different. So, if you know the prefix and suffix, so you can write without even studying also. The sex chromosome are dissimilar here. So, you will be having XX, here you will be having XY chromosome, and sex chromosomes are similar, that is it is XX chromosomes what you will be having. Usually the heterogametic males are heterogametic, whereas females are homogametic. And fourth question, write a note on erythroblastosis fetalis or hemolytic disease of the newborn. So, both are same. So, if they ask erythroblastosis or hemolytic disease of the newborn, you have to write the same. So, first, how does it happens? When there is a marriage between the Rh positive and Rh negative women, this results in the type of incompatibility. So, during the second pregnancy, the Rh child may be exposed to antibodies produced by the mother during the first pregnancy. So, this is carried across the placenta into the fetal blood circulation, and this causes the hemolysis of the fetus by RBCs and resulting in the hemolytic jaundice or anemia. So, these are the conditions what you will get during the erythroblastosis fetalis. So, marriage between Rh positive and Rh negative women, when it happens during the second pregnancy only, the Rh child, if the child is Rh positive, then he will be exposed to antibodies produced by the mother. So, this is called as the, so these antibodies which are produced in the mother, they cross the placenta and they will go into the fetal blood circulation. So, what happen? So, when they enter, they will hemolysis. Hemolysis means breakdown of the RBCs happens and causes the jaundice and anemia in the new child. Now, what is meant by kin selection? So, kin selection is nothing but all other females which are diploid having developed from fertilized eggs of, they help to raise the queen's egg, and so they contribute the queen's reproductive success and indirectly to their own, which is known as kin selection. So, it is usually seen in hymenopteran insects like honeybees, wasps and ants, and it also helps in evolution of sociality. One, define Klinefelter syndrome. So, these are XXY males and they have additional copy of X chromosome with 47 chromosomes, that is 44 autosomes plus XXY chromosome, and the symptoms of Klinefelter syndrome are they are sterile males, tall, obese, long limbs, high pitched voice and underdeveloped genitalia. So, these are the symptoms they have, that is the males cannot produce fertile sperms, that is called as sterile males. They will be tall and they will be obese and they have long limbs and their voice is high pitched and they have underdeveloped genitalia, not properly developed. The seventh one, define Turner syndrome. So, these are XO females. So, they have 45 chromosomes instead of 46 chromosomes, out of which 44 are autosomes and they have only one X chromosome and no Y chromosome is present. So, no another X chromosome is present. So, that these are called as the Turner syndrome. So, the symptoms are sterile female, low stature means they are very short, and webbed neck, that means the neck and the head are very close, and underdevelopment of breasts and rudimentary gonads. So, gonads means the fallopian tubes and the ovaries are not properly developed. So, that is called as the gonads. So, we will be continuing in the next class the name mark and 5 question. Thank you.